PD-L1 has become an essential biomarker in oncology because of its proven clinical utility in predicting which patients will respond and benefit the most from this new class of therapies called immunotherapies. The problem is that the PD-L1 biomarker is fairly difficult to interpret by pathologists. It takes quite a bit of time to interpret correctly, and multiple studies have shown that the inter-reader reproducibility for PD-L1 scoring is fairly poor. So there's an opportunity to use digital pathology and AI technology to improve both the accuracy and the reproducibility of PD-L1 scoring. So PathAI has created a machine learning algorithm to address the PD-L1 interpretation problem, and this is the focus of the AIM PD-L1 poster at AACR. The poster documents PathAI's efforts to create a sophisticated machine learning algorithm to automatically score PD-L1 for multiple clones on the market, including SP142, SP263, 22C3, and 28A. There was some really impressive work to train the algorithm that included non-small cell lung cancer samples, over 5,000 of them, and human pathologists annotated these samples and generated over 300,000 annotations, both at the tissue level and the cell level, that went into the algorithm itself. And the remainder of the poster then measures the performance of the algorithm compared to 12 human pathologists who scored the PD-L1s manually. We looked at the algorithm performance compared to 12 human pathologists who each independently scored each of the over 350 cases that were part of the test set. And in addition to each individual pathologist's score, we also generated a consensus score of all 12 pathologists, and this also was compared to the performance of the algorithm. And what we saw in terms of the results that you can see in figure 2 of the poster is mainly two things. One, as expected, we see a wide range in performance of the human pathologists. So the 12 pathologists, when you compare each of their individual performance to the consensus, you see a wide range. Some pathologists correlate very well to consensus, and other pathologists correlate very poorly to consensus. In contrast, the AI algorithm from PathAI correlated very highly with an interclass correlation coefficient of 0.93 compared to the consensus of 12 pathologists. The data suggests that this is a more accurate, more reproducible way to score PD-L1, which in the end is going to be better for patients. Another major issue facing the field of oncology and pathology in the case of PD-L1 is the fact that there are multiple clones on the market. And all 4 of these clones have received FDA approval in different indications. That's good in one sense because of the availability of therapies to multiple different disease indications. But in terms of testing, it's a problem because pathologists find this very confusing to have 4 different clones in different indications, 4 different scoring systems. And we know both anecdotally, but also more and more with published data, that this confusion leads to inaccuracy, which can impact the patient. So I think artificial intelligence and machine learning can also play a role to help standardize diversity and heterogeneity in testing, like we're seeing with PD-L1. So in the case of the poster here where we're talking about that was presented at AACR, the algorithm was trained on all 4 clones that are on market. And in the future, I think this will be very helpful to pathologists because it does sort of on the back end help standardize the interpretation.